Today we're going to see about Salmonella. This is a very complex group, contains more than 2000 species. They are typed on the basis of serotyping and the species typing. They are divided into two groups. One is enteric fever, which is a typhoid one, and depend upon the food poisoning, they are divided into the two types. Morphology of Salmonella, they are the gram-negative bacilli, which is 1 to 3 microns. They are motile by the peritrichous flagella. The enteric fever group, which is a typhoid fever one caused by the Salmonella typhi, and other groups called as the paratyphi ABC. The Salmonella typhi causes the typhoid fever. Salmonella paratyphi ABC causes the paratyphoid fever. Bacteriology typhoid fever. The genus Salmonella belong to the Enterobacteriaceae. They are aerobic in nature and they are gram-negative bacilli. They are distinguished from the other bacteria by the biochemical and antigen structure. Their clinical manifestations are headache, malaise, anorexia, coated tongue, abdominal discomfort, constipation or diarrhea, hepatomegaly, and the rose spots appear. Complications of the enteric fever. They are causes the intestinal perforation. They also causes a hemorrhage, circulatory collapse, bronchitis, bronchopneumonia, meningitis, nephritis, and osteomyelitis. The food poisoning group, which is the second one, they are non-typhoidal salmonella. This is the foodborne illness caused by the salmonella. Their transmission occurs via the contaminated food and water. They are mainly from the poultry and eggs. They are caused by exotic pets also like turtle stool. They contain the salmonella. More than 90% of the reptile stool contain the Salmonella bacterium. Here we can look for the general signs and symptoms. Diarrhea with fever, abdominal cramps and nausea and vomiting. The symptoms of the most Salmonella infection usually occur 6 to 72 hours, usually 12 to 36 hours after the ingestion of the Salmonella, and the illness lasts up to 2 to 7 days and go away without the medical treatment. Here in the day 1, the stool sample are collected in the selenate fecal broth. They should be intubated at 24 hours. They are used for the isolation of the Salmonella from the contaminated specimen. In day 2, the culture on the psilocybin desoxylate agar should be done. Xylose fermentation, lysine decarboxylation, and the production of the hydrogen sulfide are the primary differentiation of the Salmonella from the non-pathogenic bacteria. On the third day, they observe the colony. They suspect if you have the black colony. They discard if no black colony occurs. Urea test. They discard if the positive urea. Suspect if the negative urea. Microgen and the sensitivity test is also done. In the day 4, the lysine decarboxylase gives a positive sign. Glucose will be present. ONPG, Voges prosecure. These are the tests that we are doing, and they will check for the ornithine decarboxylase and the mannitol. It should be present. Indole should be negative, and xylose should be negative. Urea hydroxylase should be negative, and the tryptophan deaminase should be negative. Observing the sensitivity test, they should be sensitive to the ceftriaxone, cefuroxime, and trimethoprim or sulfonamide. They should be sensitive to all of these drugs. This salmonella should be resistant to ampicillin, amoxicillin, chloramphenicol, and the penicillin G. The result is dispatched to the ward. Treatment. The electrolyte replacement, that is the sodium, potassium, and the chloride ions, lost through the vomiting and the diarrhea. So, the routine antimicrobial therapy should be given. Control if the infection spreads from the intestine to other parts of the body. Prevention. Not keeping the reptiles as pets, particularly if you have the younger children. Proper hand washing should be done. Not serving any raw meat or egg. Prevention from all stages of the food chain, agriculture production, processing, manufacturing, and the preparation of food should be done. Thank you.